Okay, so hi everyone. So today we are being presented to an introduction to biophysical concepts, and so by Joseph Kuhi. So please join me in welcoming him with a warm round of applause.
Yeah, as she said, I'm Joseph. I'm a third year biophysics specialist student here at the University of Toronto. And today I'd like to talk a bit about biophysics because, as I know, anytime I've ever mentioned it to someone, they're very curious what it is, whether they're doing biology or doing physics. It's a very sort of mysterious field, even to those who are studying it sometimes. And to go through it, I'm going to start at sort of a larger scale, and I'll go sort of smaller and smaller, observing instances of biophysical or physical concepts in biological contexts. Of course, first we'll start with sort of macroscopic biophysics. This is sort of inherently classical in nature, right? Any sort of amalgamation of bone and muscle can be uh, broken down into, tension strings and rigid beams. We can make a free body diagram of kind of any fundamental movement of a person. And this can definitely be useful when you're conceptualizing or trying to understand how a person or a thing may move, particularly in biological contexts, right? But it can be very interesting when you use it on some more less than intuitive systems. And one of them I'd actually like to bring up is the biophysics of the human ear. The human ear is a very interesting thing. It kind of has a lot going on here, and so I'll sort of describe the general process of how it operates. At the far sort of left here, we have the outer ear, and through there, sound waves will sort of permeate and they'll come all the way inside, and then they'll hit the eardrum here. And this is sort of an elastic surface. And what happens is it'll actually resonate back and forth in tune with the sound waves. And then that's when we have the middle ear. This is where it comes into play, right? There are three bones here, and these three bones actually can sort of flex and bend with one another. And as the eardrum oscillates back and forth, it actually moves those bones. And the bones will slowly press back and forth on the cochlea, which is the inner ear. Now, inside the inner ear, it is entirely pressurized. And so all of these movements back and forth on this sort of pressurized surface create pressure waves, which are sent all throughout the structure. And inside there are many various hairs which will actually detect the movements very minutely, and then it'll carry those as electric signals or sort of as chemical signals to the nervous system, which then goes to the brain. And immediately we see sort of some classical considerations. If you've sort of been paying attention or if you've taken some of the classical physics, you might be able to associate particularly the middle ear with damped harmonic oscillators or driven in damped harmonic oscillators, as we might be able to say. We can sort of associate that eardrum and its movement induced by the sound waves as a sort of driving component. We can associate the inner bone as the spring, right, which has its own sort of internal constant depending on how it interacts with these sorts of things. And we can consider the dampener of all the various aspects, the muscles on the inside and the resistance of the pressurized cochlea. And another small thing I'd actually like to mention is down here, just below where it's actually pushing, there is a little opening. And what happens is as the bone presses inwards, this actually presses outwards. Because it wants to maintain its pressure throughout the entirety of the cochlea, it needs to sort of release or kind of have an exit for that pressure that's being applied to it, right? And so it has that round window there, which will actually sort of bulge out as the other pieces in on the oval window there. Now, it's classified as a sort of pseudo-damped driven harmonic oscillator, which is shown by this equation here. The reason I say it's pseudo is because it's not constantly damped. It's only damped momentarily or for as long as there are sound waves sort of permeating or affecting the eardrum. I'd like to momentarily talk on the sort of harmonic oscillator equations. Here we have the damped and driven. In this case, we'd be considering an underdamped harmonic oscillator equation. And this is because as it continues to oscillate after the sound has already hit it, right? An overdamped would just sort of end before any sort of oscillation, same with a critically damped. And so we want that continued oscillation. We want that continued effect on the cochlea to ensure that we're still hearing things, right? And so it's represented by this equation on the left. Of course, we have m mass times acceleration, and then plus kx, which is this is the equation of motion, right? For a spring functionally, or a sort of any basic harmonic oscillator. Mass on a spring is a classical example. And then we have the b times velocity. That b is that sort of damped constant, that damping effect that's being applied to it. And over here we have the damped and driven. And as we see here, there's something considered a steady state regime where it'll stay sort of constant. And that's because this new term we've introduced, the F naught cos omega t, is going to sort of counteract the other one and prevent it from going all the way to zero. And so it'll continue in this state as long as the driven factor, the factor that's continuously applying force to this system, is there. So if you hear a note constantly, for example, if you're hearing a sound as one sort of pitch and you're constantly getting sound waves into your ears, then you will sort of observe this steady state regime in how it's oscillating. But the moment someone stops talking, it'll go to that damped, and there's no longer a driving. Um, a reason this can be very useful for considering these systems is because it gives us insight into the nature of how they work, but also how we may be able to apply any sort of considerations to them. Theoretically, think that there's somebody who might have sort of an ear that is not working as well as it should. They're not hearing as well as they should be able to. And so if you consider this equation, you might understand that somehow, somewhere, it is being reduced. Somehow, it is sort of not transferring that energy as much, and the intensity of this oscillation is not as strong as it could. Of course, one common thing is something like earwax. A buildup of a lot of earwax is a problem and can hinder the eardrum's ability to move back and forth. The same can be said here in this aspect, or in how it's pressuring on sort of the cochlea itself, if it's not pressurized as a major issue. And we can consider these in the equations, right? We can associate that spring constant K with the actual movement of these bones and the eardrum. And in doing so, we have a better understanding of how exactly, sort of the stiffening or the blocking of here of the eardrum with something like earwax could ultimately result in a reduction in the actual intensity of the simple harmonic motion. We can also conceptualize sort of an idea on how to treat something like this, right? We could, of course, change that spring constant, increasing it by removing something that's interfering with it, or any sort of issues that may be within the inner ear here with those bones. Also, in terms of the driven factor, if it's not driving as hard as it may be, or perhaps it's something more internal that can't be directly sort of affected, we can amplify it. And that's what something like hearing aids are used for, right? Functionally, they function as amplification, and they're sort of increasing that driven on the right of the damped and driven equation. And in doing so, it amplifies, the sound waves that are coming in. It imparts greater force on the eardrum, right? And so it's sort of overcoming that difference in a reduced K or perhaps an increased B. Any questions? Or okay. So that's the human ear. I'd like to go to another sort of less common example, but one that's very interesting to me, which is hemodynamics. This is sort of the hydraulic movement, or the pressurized movement of blood flow throughout the body. Of course, it starts at the heart, which pumps it all throughout, right? And it goes, there's the deoxygenated blood, which is entering the heart, and there's the oxygenated blood, which is leaving the heart, and it's sort of going all throughout. It's going towards those capillaries where it's getting blood, and it's sort of being pumped out. This is a very, very important process, as hopefully you know, because everyone needs to sort of live. And as we can conceptualize it, we can consider it as a sort of complex hydraulic circuit, right? Where it's sort of carrying these nutrients all across the body. And as its main role is functionally to sort of transfer that oxygen, it's very important that this sort of hydraulic circuit operates at a very good efficiency. One way to sort of calculate or consider the efficiency of this is using the Fick equation, or the Fick principle, of cardiac output. So cardiac output is in liters per minute, and it's functionally a description of how much blood the heart ejects per minute, right? How much blood it's actually going through or pumping through the blood. Considering we're dealing with what is constant volume of blood, or hopefully a constant volume of blood, um, we can sort of apply our sort of observations of the blood coming in and the blood coming out to understand how efficiently the blood is actually functioning, right? We have the consumption of oxygen per minute over here, which we can dictate by just how much someone breathes in, right? The kind of air they're breathing in. And then we have the concentration of oxygen in blood leaving the heart and concentration of oxygen in blood leaving, sorry, one of those should be entering, this one should be entering the heart and leaving the heart. Um, those, of course, can very easily be measured by taking blood before and after it enters the heart in the circulatory system. And so this is not a very difficult way to understand how exactly, how sort of strong an individual's heart is, how good it is. And this is a very important sort of thing to do, particularly when you're diagnosing cardiovascular disease or someone who might have hereditary cardiovascular problems, right? If you have a weak heart or if you have something that is actually blocking your consideration of this equation of what you're getting from it is very important. One of the reasons we actually consider this is because of Fick's first law of diffusion. He was really busy, and as we can see, diffusion is the fundamental way in which blood manages to get oxygen to different parts of your body. Right, it's diffusing it into the cells, into the surrounding, and it's intrinsically linked as a concept to this. And if we think about our sort of cardiovascular or cardiac sort of constant, uh, we can conceptualize it in the context of a sort of diffusive range, right? We have J, which is sort of the diffusive flux, which is the amount that's being diffused over an area per unit time. And then here we have the dC over dx, which is going to be our concentration, right? Or the difference in concentration between two different sort of diffusive, what's sort of diffusing from one side to the other. And then we have the diffusion coefficient. The concentration gradient, sorry, the concentration gradient is going to be dependent on the concentration of an individual molecule on both sides of the sort of diffusive membrane, right? And more often than not, the more stuff we are getting, the more higher concentration we have on one side, it's going to create a greater concentration, right? If there are more molecules sort of, here and there's less here, it's going to go over, right? But if the blood, for example, is moving very, very slowly, or it's not moving as efficiently as it may be able to, it's spending more time in one place or in one area of the body, and thus is not diffusing as much as it could necessarily. You want that high concentration as often as possible. And so your body is going to ideally want to pump as much blood as it can efficiently across the body as regularly as possible to ensure that your oxygen concentration in your blood is actually high, so that this diffusion gradient or this concentration gradient, sorry, is greater, and thus the diffusion flux is greater, ultimately resulting in more oxygen getting to what it needs to be or where it needs to be in your body, which is significantly healthier process for a person, right? The more oxygen you have access to, the more your cells and everything within it can use that to keep you alive as efficiently as possible. Yeah, so this is sort of considering both of the equations together, right? Sort of how we can use that cardiac output in the context of the sort of diffusive gradient, right, or the diffusive flux. Of course, I'd be remiss if I didn't talk a bit about biological imaging. This is a big part of biophysics, right? Here's an example. Of course, we have X-rays. They sort of have a source of X-rays, which are exposed to a body, and then based on the sort of density of the various parts of your body, they absorb those X-rays to varying degrees. And then, as you see, sort of here's an example behind the person, there's an X-ray film, which will create sort of an imprint of the sort of shadows and light, as it were, of the X-rays going through the person. This imprint will give you an indication of bones. It'll give you an indication of all kinds of things. It's usually better when dealing with denser things, which is why it's very common for bones, right? Because they'll absorb it a lot more than the surrounding muscle tissue, and they'll give you a good indication of how that's actually operating. Another biological imaging I like to mention is MRIs, magnetic resonance imaging. I'm sure, I don't know, some of you may be familiar, especially if you've done some chemistry. Um, it's a very complex process. I will say this is a very significant oversimplification of it. But fundamentally, it sort of induces a large magnetic field, which will align a lot of the protons in the water that you're made of, right? You have a lot of water within you, and all those protons within that water will align accordingly. In this case, it's actually a 1/2 spin, sort of the quantum spin of these things that will align. Um, it's a very quantum mechanically involved process, I will say. Then what happens is radio waves will sort of induce excitation, and they will sort of flip some of the protons. They will change directions, or they'll change their spin, and then afterwards, when it's sort of released, when the radio waves are gone, they will reorient themselves. In reorienting themselves, they release the excited energy, and the excited energy can then be measured and used to dictate what exactly the makeup of those specific things are. In a grander scale, when you're using a macroscopic system and you do this, you can produce an image based on all of the energy that's released. And that's how MRI images are utilized. Next, I'd like to discuss microscopic biophysics, or the concept of uh, microscopic systems, or microscopic biological systems. Um, Fundamentally, they're very chemical in nature. You're dealing with a lot of chemicals. Chemicals are everywhere. And at this really small scale, thermodynamics is sort of the driving rule and law of everything that happens. Free energy exchange is the main principle of any given interaction, and is fundamentally the most imperative aspect of anything that goes on. And at this level, the main thing that does functionally everything is proteins. Proteins themselves are sort of molecules. I will actually, what are proteins? Yeah, proteins are complex molecules. They're made up of a singular chain of what are called amino acids. Your body has 21 different amino acids. It's chosen 21. We don't know why. There are more, but we picked 21. And based on this chain, it'll actually take a certain conformation or a certain shape, and this shape will move. It'll interact with other molecules, and it'll do sort of jobs, as you'll see in a bit. Of course, I'd like to touch a bit on amino acids. There are 21 different ones, right? They're all synthesized based on your DNA in a very interesting process of DNA translation and transcription, which has all kinds of very confusing things involved with it. But they all have various different properties. Some of them are charged, some of them are neutral. This also depends on things like pH and the environment they're in. But as you'll see within the cells, it's actually a very neutral or relatively neutral pH, and it's because it's very, very similar to water, which is very integral. So how proteins take shape? As you see here, these are some proteins moving. On the left, on your left, on the left, we have a protein that's sort of conforming. It's taking a particular shape based after its synthesis, right? And on the right here, we actually have a series of proteins, a complex that is moving and actually acting within the cell. This is what it would be visualized as. There are two big or primary driving factors that contribute to the movement and the conformational change of proteins, and those are steric interactions and hydrophobic effect. Steric interactions are sort of non-binding interactions between molecules, and it's the sort of repulsion and interaction of the sort of surrounding electrons or electron orbits of individual molecules. Of course, here we have butane as a sort of example, a sort of classical example. Here we have this conformation in which both C bonds or both sort of C strictures are on the same side. And because they're relatively close to each other, these sort of surrounding hydrogens don't like being that close necessarily, right? Their sort of electrons don't want to be as close as that. And more than anything, they want to go into what is an energetically favorable state where they are separated or sort of as far as they can be from each other in this bond. And so they will actually sort of rotate along that bond into this much more stable state. It's not impossible for them to be in the other state, but it's less energetically favorable. And as they stand, they're significantly more likely to change to the stable state. Yeah. Next, I'd like to talk about the hydrophobic effect. Amino acids inherently have something called a hydrophobicity, which is how hydrophobic or hydrophilic a molecule is. If it's hydrophilic, it really likes water. If it's hydrophobic, it doesn't like water. And these are determined by their polarity. Water molecules, inherently, as they are two hydrogens and, actually, I should probably draw this. They're two hydrogens and, horrible drawing. They're two hydrogens and they're an oxygen, right? This inherently is not a symmetrical shape, and because of that, there's an induced dipole, or some part of it's more negative than the other, which is more positive, right? Um, this inherently means that it actually likes being near other things that also have a dipole, other molecules that have that sort of dipole moment, or some sort of difference in electronegativity across its molecule. And so here we see how there's sort of a gradient. It's not a hard and fast rule. Different molecules have different levels of how much they like to be near or don't like to be near water. And a lot of it is dependent on their sort of how their polarity. You'll actually notice over here in the sort of higher region of something that's hydrophilic or really likes water, these are all charged. Many of these are charged, and it's because they're very attracted to that polarity. They're very attracted to that difference in sort of charge, and they really want to be near it. And then things usually further down the hydrophobic spectrum, more hydrophobic in nature, are significantly more neutral and much more evenly distributed in their electronegativity. And what this ultimately results in is, given that proteins, when they're synthesized or they're created, are within a very aqueous environment, a very water-focused environment, as you can see on the left here, they quickly begin to take shape or form where all the hydrophilic molecules will sort of spread outwards or want to gain as much access as possible to the surrounding water, while all the hydrophobic molecules or the hydrophobic amino acids will sort of bend inwards, trying to escape the water, and will kind of clump up together. And in clumping up together, they create structure, based specifically on the sequence. So here we see, initially there's this sort of isolated protein, and then when it's in an aqueous solution, all those red hydrophobic regions or those red hydrophobic amino acids bend in on one another, and they sort of try to escape the surrounding aqueous solution, while all the other ones try to sort of stretch out and have as much access to the water as possible. And so based specifically on the sequence that is synthesized for a protein, when it is sort of let free within the cell, it'll take its conformation, or it'll just take its shape naturally through this sort of process. And this process, of course, is driven primarily by energetic favorability, right? The sort of Gibbs free law, or Gibbs free energy equation, sorry, is the driving factor behind most of this. On the right here, we can actually see a sort of potential energy or free energy plot here, and we see that down here, the bottom of this well is the most ideal conformation for this protein. It really wants to be down here, and it sort of falls down this gradient as best it can into this position, because while it is here, Gibbs free energy is reduced as much as possible. It's at that highest entropy or the lowest enthalpy, and in doing so, it is ideally satisfying the thermodynamic principles. When it is out here, it's significantly less stable, and there are some smaller pockets, but usually it will sort of fall out of these as it moves throughout the cell. This, of course, is the driving factor for both the hydrophobic effect and the steric interactions. All these molecules are trying as much as possible to get into the ideal sort of situation, the ideal shape they can be in, and in doing so, they're actually creating shapes that are perfect for functioning a certain role or a certain sort of job, as it were, within the cell. An example of this I'd like to bring up, as sort of previously mentioned, is ATP synthase. ATP is amine triphosphate. It's a very important molecule, or a very important chemical within the cell. It's responsible functionally as sort of an energy source. And so this creates that molecular energy source within your body. It's made up of 29 different proteins. So as you can see, proteins don't necessarily exist holistically individually, but they exist as very, very complex systems that can come together and interact with one another. Here you see all of these different colors are different proteins here. There's some that focus as backbones and some that interact over here. And this conformational change it's taking is actually within the context of other molecules I will describe, as you can see sort of on the left. But it's all sort of abiding by that free energy sort of exchange as much as possible. And you might be wondering how exactly it's moving. It needs some kind of power, it needs some kind of energy. Once it gets into that shape, it's not just going to keep moving infinitely, right? And actually, your body does something very, very smart. It uses a proton gradient. And so here, there is usually a wall, as you can see over there, a wall of lipid membrane, or a membrane wall that will separate this side and this side. And what happens is there's significantly more protons here than there are over here. And these protons, of course, naturally want to leave. They want to go where it's less concentrated. They want to achieve that sort of highest entropy possible. They want to minimize the free energy and they want to escape. But in order to do that, they have to go through ATP synthase. And of course, ATP synthase has a way to let it out, but it's not going to let it out without doing a little work first. And so as it comes in, it uses the energy produced by that proton going from one side to the next as it sort of turns its potential into a kinetic while it's moving to actually power its movement. So as it moves through, it sort of pulls this along and helps it move along. And what this reaction is doing over here, this is sort of powering almost as a wheel or an engine that powers this interaction up here. And the interaction up here is taking ADP, which is adenine diphosphate, and it's adding another phosphate to turn it into a triphosphate. And so it'll bind to both a to a phosphate and adenine diphosphate. And using this energy, it'll pull them together and it'll actually induce that reaction. It'll catalyze it. And it's sort of using this proton gradient as something that powers these sort of almost mechanical or catalytic source for creating these new energy sources. And then it'll release ATP into the cell. And then somewhere else, something will grab ATP and it'll use the energetic favorability of breaking up ATP to power something else that it's doing. And so it sort of repeats all throughout the cell as much as it can. This protein actually is present within the mitochondria, and this sort of inner side should be the inside of the mitochondria, and I believe outside, or there's two layers of the mitochondria. It has sort of an outer layer and sort of an inner layer, and the inner layer is very proton rich, the outer layer is not. And so that's why they consider the mitochondria the powerhouse of the cell. It's responsible for creating one of the most important molecules in it. Of course, biochemicals have imaging too. You need to understand and sort of see their structure as much as possible. NMR spectroscopy is actually very, very similar to MRIs. The sort of function or imaging of it is a similar process functionally, but instead of at a large scale, it's doing it at a very, very small scale. And so you get these much more defined kind of graphs where you can tell where individual molecules may or may not be present, depending on how shifted they are in their resonance. And of course, it's very useful for determining particular chemicals. This is not just within biochemistry. Any sort of chemistry will have NMR spectroscopy. It's a necessary aspect of sort of being able to understand what a specific molecule is made out of. And that's very, very useful because if you know what a molecule is made out of, you know how to engage with it, which is something very necessary if you're developing something like medication or drugs, because functionally those are supposed to attach and interact with proteins and molecules and change them in a specific way. And so by considering their shape and considering how they can form or change, you can do that. There's also something called X-ray crystallography. I really love this one. What they'll do is they'll isolate proteins sort of from a cell, and then they will grow a crystal out of them where they will align into a physical crystal, and they'll shoot X-rays through it. And based on the diffusion of those X-rays, based on the diffraction, how they sort of bounce off and they'll create sort of scattered dots, they'll actually translate that into a structure for the protein, which is very, very interesting. It's very math intensive, right? There's a very complicated computer and a very complicated algorithm for how it basically takes what are these individual points and translates them into positions for molecules. Finally, I'd like to talk a bit about quantum biology. The field is sparse, I will say, in terms of quantum biology. Inherently, the systems of biochemical interactions are quantum mechanical in nature, right? Electron orbits and the interactions of electrons in bonds are inherently, probabilistic and not deterministic. And so quantum mechanics is sort of ever present in that nature. But systems outright using quantum mechanics is not very commonly present, and it's significantly more theoretical. That's, of course, because quantum mechanics is very difficult to observe in a biological system, given how much just kind of stuff is around, right? it's much easier to observe quantum mechanical systems when they're sterile or sort of separate, not with all these other molecules surrounding it and under a microscope or something. It's much more difficult to observe. And as far as we're aware, there's very few instances or even possible instances of these systems intentionally engaging with quantum mechanics. One possible one I'd like to discuss is a theory for point particle mutations devised by Lodewin back in, I believe, 1960s, which says that quantum tunneling may be responsible for some mutations in the genetic sequence of living beings. The genetic sequence of living beings, of course, is sort of a series of molecules that have, I apologize. It's a very odd thing to explain. But the genetic sequence are sort of these two chains of molecules, and they're both sort of bonding to each other back and forth. And there's only 4 possible molecules they can be. And of course, they're bonded in pairs. So A with T, C with G. They have to bond together with those specific molecules. And this creates a sort of code, as it were, which when read a specific way will produce proteins, or it'll produce RNA, which will then produce proteins. And in a way, you can consider it as sort of a code of the human being, right? When it's read specifically, it produces information, or in this case, molecules. But it's very particularly bonded across these 2 chains by hydrogen bonds. And of course, hydrogen is just small enough to do some quantum mechanical things. And an example of that is that certain individual mutations were observed where hydrogen would misbond. It would accidentally not bond between the 2 it was supposed to, but would actually bond to one just over. And this, of course, as Lodewin attempted to explain, uh, theorized was a quantum mechanical process. In this case, he traced it up to quantum tunneling. So conceptually, quantum tunneling, of course, is when you sort of bypass a potential barrier by going from one initial state to a new state, and you sort of go through this potential barrier from one potential to another. And in this case, it would be the potential of that initial hydrogen bond to the potential of this sort of new mutation or misbond. In that this hydrogen, or this single photon in this case, would actually sort of pass through that potential barrier. Over here, we can see sort of some of these, the associative conformations or the associative bondings that are possible with the hydrogen and their associated sort of energy. And in these individual cases, I believe they somewhat, not entirely align with this, but you see that some are more likely than others. And if you can kind of almost go through 2 of these potentials to that AT1 or that AT3, that sort of slight misbonding within the molecule, you can almost induce what is a minute mutation. I will, of course, preface this is not technically proven as it stands. It's theoretical work as it exists because it's something very hard to observe. You can observe those mutations, but you can't necessarily observe how they were created, right? That's happening deep within the cell over the course of some time, and so it's very hard to see. Yeah, so as it stands, very theoretical, but very interesting because it does mean there's a possibility, you could attribute some degree of evolution over the course of however many hundreds of thousands of years to quantum tunneling. And that's the end of my presentation. Thank you very much. If you have any questions, feel free to ask them. I will be here until they kick me out, probably. And thank you.
Yeah, does anybody have questions?
Yes.